Yes, the first presenter for FAHN research is Dr. Sunita.
Hi, everybody. I'll just get my slides together so that everyone can see. Okay, you should be seeing my screen now.
Yes.
All
Yes.
good? Perfect. Okay, great. So this is actually quite exciting because this is the first time I think that we've had 2 back-to-back sessions on FAHN at a family meeting. The last family meeting a couple of years ago in Charleston, it was a small, intimate gathering of friends where we discussed potential research, where things were going, but really didn't have as much to share as I will be sharing with you today. So I'm really privileged to be part of this community, and I really thank the organizers for inviting me to speak today. So just to give you a little bit of background as to how I got into this. So I'm from Ottawa, Canada. I'm a clinical neurologist, a pediatric neurologist, and I happened to come across my first patient with an NBIA about 7 years ago now. I didn't know much about NBIAs at the time. It seemed like a black box to me. So I happened to be at a meeting that I went to in Spain later that year, where I met with Dr. Hayflick and had some discussions about this patient further. After which I was invited to go to my first TURCON meeting back in Germany in 2015. And that's when I really got into deeper discussions about where I could fit, where there's a hole or a missing link in the NBIA world, and where as a clinical neurologist could I fit to help patients and families all over the world who are affected by FAHN to move forward in terms of understanding treatments, building a community, etc. So after this, I was able to go to my first NBIA scientific meeting, and following this is when everything else that I'm going to show you has come to take place. I will be just getting rid of my chats because that is not anything anyone needs to see. So I will just give me one moment, please. Okay, so in general, all of you know fatty acid hydroxylase associated neurodegeneration is a neurodegenerative condition that tends to affect children, but affects various neurological systems, including movement, which includes spasticity, dystonia, ataxia, all coming from different parts of the brain, speech and communication, learning, affects people in having seizures, their vision, feeding, and bowel and bladder function. We know that it's different from the other NBIAs in terms of where it exists. So if this is a cell, within the cell, you can see that there are regions where PANC2 is responsible for PCAN, where WDR45 is responsible for BPAN, and where there's lots of research going on in these different areas. We do see that FA2H, the gene responsible for FAHN, is in a completely separate region. And I'm going to go into that a little bit further throughout the talk. So one big question that's been brought up over the years by many families is, why is FAHN called so many things? Why do some people refer to it as a leukodystrophy? Why do some people refer to it as an NBIA or a spastic paraplegia? And I want to say that it's all of the above. And so leukodystrophy really means that it's an inherited disorder, so disorder that affects genetics in some way that affects predominantly the white matter of the brain. So the myelin regions of the brain, which is the insulation of our nervous system, of our nerves. NBIA, as we all know, is evidence of iron accumulation in the deep structures of our brain, like the basal ganglia and the substantia nigra. And these areas are extremely important for different types of movement. And spastic paraplegia is, there are so many different spastic paraplegias out there, and each of these is a clinical entity to start with. And over the years, we've been finding that there are different genes that are responsible for various different spastic paraplegias. And sometimes one gene is actually responsible for multiple different types of spastic paraplegias. So FAHN and SPG35, yes, they're the same condition. SPG35 was discussed and discovered by those that were studying spastic paraplegias, but it's the same FA2H mutation that causes SBG35 as is in FAHN. So FAHN has elements of all of the above, and I think this is really important because we can utilize these different the understanding of these different constellations of diseases in order to better understand FAHN in more detail. And we can also use the knowledge that we gain from FAHN to better understand these other conditions in more detail. So it goes back and forth. We use the term FAHN because it includes all of these patients that have two different mutations on FA2H. So we know this is autosomal recessive. What that means is that one mutation has to come from one parent and the other has to come from the other parent. And there have to be two of them on this gene in order for FAHN to occur. So what I've been focusing on, first of all, as a clinician, is a natural history study for FAHN. And this has been funded, fortunately, by the NBIA Disorders Association a few years ago. And as you've heard from Dr. Sid and from other speakers, it is really important that we have natural history studies for rare diseases. And it's important that we can follow patients through their natural course. Not only do we get to see what happens at different stages of the condition, whether or not they're on different treatments of their own choice, but we can also identify different windows in time where, if a therapy comes to place, where it may be most useful. This can also help if we understand where different changes are happening in a child or an adult. We can understand how to guide not only the patient and their family, but also their clinicians as to what needs to be looked for ahead of time so that we don't miss any opportunities to optimize health. It also helps us understand the incident. If we know what the incidence and prevalence, so we know how many patients or how many families are affected every year, or how many there are in the entire population, then when there is a treatment that is available, then we can actually advocate for this treatment to be allowed to be put into a clinical trial, because we can give actual numbers within different populations. And then we can also find sites around the world that may be able to contribute to the clinical trial that comes into play. So as you all know, the first clinical patients that were identified in 2008 by Catherine Dick. And these patients were found that everybody had normal development early on, followed by changes in the way they walked. So their gait was becoming spastic. I'm going to use these terms, and I'm going to explain these terms a little bit better because I'm sure you're all familiar with it, but spasticity is something that has to do with increased tone that is dependent on how quickly that limb is moved. And so things like spasticity can get worse with quick movements, but also can be worse in certain positions. And they tend to happen, spasticity is seen more during daytime wakefulness hours, but can also be present at night. So these families were found to have rapid progression of spasticity involving first their lower limbs, then their upper limbs, followed by dystonia, which is another tone abnormality, which is treated differently, but it's got to do with abnormal posturing or muscles that should be relaxing against those that are contracting. Instead, the contracting muscles and the relaxing muscles are contracting at the same time, abnormally, because of signals that are being misfired from the brain. And so in combination of spasticity, dystonia, there's also regression in learning, as well as problems in the cerebellar function, which means that there can be problems with tremor or ataxia or instability, whether sitting or standing or trying to walk. There was a larger study that came out in 2019 from the German group by Dr. Ratte and his group. And in this family, they found 19 patients who were not of which a few were previously described, from 9 different families. And again, they found that motor issues were the first sign around the age of 4. And by the age about 7 years after having had symptoms start is when ambulation was lost, or inability to walk anymore. And when they looked at these families, they looked at both MRI findings, they looked at the different clinical findings, and they looked at the MRI findings looking at the white matter changes. Were there changes that looked like there was iron? Were there changes where it looked like the brain matter was actually becoming less volume compared to normal control patients at that age? So they found that there were changes in all these things. But what I feel that we needed to do further was actually to look at the timing of all this, because this can really influence future therapies. So what we did was we looked at an international cohort, of which all of you have been a part of, clinical and MRI characteristics of children and adults who have been diagnosed with FAHN. And we wanted to look at the specific gene mutations that were associated and look to see whether these are in important regions in the gene. The secondary things we wanted to look at is what was the earliest symptom that happened and when were the earliest findings on neuroimaging. So to do this, we went through the NBIA Disorders Association, through Turcon, through NBIA Canada, and also through collaborators who have written previous reports on FAHN to see if we can get updated information or new information on families around the world. So we had lots of phone calls, lots of emails. We went to laboratories, and we really tried to find as many families as possible. We did both clinical record. We had collaborators look at their clinical records, or we had people send us their clinical records directly to us with consent. We also did some Skype interviews or iPhone interviews to try to see if we can actually examine families live over the screen as best as possible to complement what was in the medical records. And then we put this all into a REDCap database. And the REDCap database was built by looking at all the previous known information. So this is important because this actually affects how we're going to move forward with things. So when we looked at all the different milestones that were out there, we divided them up according to how we felt we're going to be able to gather information most accurately. So we looked at motor milestones, such as what was the maximum that someone is able to achieve, when things started to change, did your child walk? If so, when did they walk? When did things change again? Did your child need support, whether that was a walking aid or AFOs, or did the child need a walker or a wheelchair? And if any of these were marked yes, we wanted to know when that happened. And this is just an example for motor milestones, but we did the same thing with fine motor, with speech, with various other regions. So this is what we found. So we studied 34 patients. 7 of those patients have been previously reported in the literature. And we got updated information from either the patients themselves or from collaborators about these 7 patients. We had 77 other patients from the literature that we also looked at in detail and put all of the information into the same clinical reporting form. So currently, when we looked at all of this, the age of the average patient was 17 and a half. Females made up just over 50%, and the age of onset of any symptoms was at a mean age of 4.5. We had a good representation from all over the world, and we divided this up by ethnicities rather than race, because we want to know there are differences depending on how we get this data. So we looked at where all these families and patients came from. So when we saw this, when we got this information, we wanted to know how old was it at start? How long did it take before diagnosis was made? And there was a delay of diagnosis of 9 years. And there's various reasons for this. First of all, FA2H is almost on all the different panels out there, genetic panels. But as you all know, genetics has changed a lot over the last decade. And now some of these families are being diagnosed through exome sequencing or genome sequencing. Previously may have been done by panels, but if the diagnosis was being attempted to be made before FA2H was even on a panel, then of course there was going to be a delay to diagnosis. So many families were given the diagnosis of cerebral palsy or spastic paraplegia or many other things for many, many years and treated as such. And this is an important thing to note, because treatment can be even symptomatically can be very different for FAHN than it would be for a child with cerebral palsy. So when we looked at symptoms, we saw that difficulty walking was the most common thing. And of that, we tried to divide it further as to spasticity or ataxia, but these things are best figured out if you actually see and examine the patient in front of you. And sometimes there can be subjectivity depending on who examined at the beginning. Therefore, we can't be too clear about what the actual finding was there. But all children had toe walking that seemed to be asymmetric with difficulty walking. When we compare this to the 77 patients in the literature, we get a very similar finding, meaning that there was 92%. And of those that were reported in the literature, many of those seem to be having spasticity or ataxia at the onset of the symptoms. Here, I find this to be a very important slide because what you look for is what you get. And so our study of the 34 is in the blue, and all of the literature that we compared to is in the purple. And as you can see, there is a lot of consistency as to what percentage of patients required a G-tube, or how many patients had problems speaking. But when you look at this further, you can see that we have found that bowel dysfunction and bladder dysfunction is quite a bit higher than what is reported in the literature. And that could be because we specifically asked for these findings where they might not have been asked in the different case reports. As well, we found that things like dystonia or movement disorders, visual dysfunction, changes in learning, and changes in swallowing were big differences in our study versus literature. And this is extremely important because this is going to affect how we actually care for your children or for your family member. When we looked further at the type of visual dysfunction, we found that there are changes such as optic atrophy, meaning thinning of the optic nerve, changes in the way the eye movements are in terms of shaking, or needing to actually move your head to have your eyes move. All of these have to do with different pathways that may be affected and are actually important to be able to separate, because that could tell us what we need to look for further in terms of what's going on in FAHN. So the next set of slides I find are extremely important to a natural history study. And so I'm going to walk you through them, but they're each going to have a very similar look to them. So what we wanted to know is, of all our patients that we were looking at, how many needed a walker, but then more importantly, when did a walker need to be used? And it seemed like after 2 years of difficulty walking, so the symptom onset, any child who needed a walker at that point. And furthermore, 4.5 years after symptom onset, 50% of our families or children had to be using a wheelchair. So this is similar to what's seen in the literature, because in the literature, the case studies found that it took about 5 years from symptom onset to being in a wheelchair. And this is an extremely important time point for all of you. And when we look at them individually, we found that each of these families, each of these children, the yellow line says how long they were in a walker from the time of starting a walker to when a wheelchair started. And you can see that this line really ranges between 1.5 years and about 3 years after symptoms have started. We looked at the same thing to see how long does it take for swallowing dysfunction to happen. So swallowing dysfunction was noted in about 2.5 years again after symptom onset. And this is really important because we need to know, when should we start assessing for this? When do we need to start discussing this with our families? And same thing with speech. When is it difficult for speech? When are difficulties with speech starting? When should we be considering speech language pathology involvement? When should we be considering augmentative therapy to be instituted? And bladder and bowel function takes a huge toll on everybody, as we know. And this is also very important because we need to be looking at this way ahead of time to prevent these problems as much as possible, because these issues seem to be happening about 9.5 years after symptoms onset in half of our families. So if we had to put a timeline together, and I haven't put numbers here in terms of ages, but early life, as you all know, is quite normal, followed by motor regression at about the age of 3.5, 4. A couple of years later, most children needed some kind of walking aid. And soon around that time is when problem speaking and swallowing happens. Soon following this, there are learning changes that are starting to happen, affecting school performance. And soon after this, the child usually needs to be in a wheelchair or have full support or mixed support. So a few years, about 5 years after all of this is when bowel and bladder impairments seem to be taking effect, and where there seems to be after this a slow progression, but all of these symptoms do not seem to be reversible. In addition, we do know that family patients have seizures. We do know that visual impairment is important, and we need to know more about the cerebellar findings, such as tremors. So these are things we need to look for in the future. So all of this leads us to looking at how should we care for families, and this hopefully will help us now write a clinical guideline for FAHN. So we know that there are very important players in terms of taking care, and there are many players. And so we want to also be able to have families not have to visit everyone at 10 different visits, but try to also amalgamate these as much as possible. So we know that physiotherapy, occupational therapy, speech therapy are very important early on. We know that eyes need to be checked by optometry or an ophthalmologist. Vaccines need to be updated so that we can make sure that you're not putting yourself at risk for any respiratory or other illnesses. Nutritional status, feeding studies need to be instituted early because we know that nourishment is really important in all neurodegenerative conditions. Dental health is important. Learning assessments have to be advocated for early. Bone health, things like scoliosis, hip health, have to be looked at by orthopedics and bone health specialists in endocrinology. We know that epilepsy can happen, and movement disorders happen. So neurologist is really important in this part of the care. And we do know that bladder and bowel effects in the teenage years happen quite frequently. And so urology and gastroenterology involvement may be important at that time. So the next part we were looking at is more about the iron and the whole iron story as to whether or not iron is actually important in FAHN, and if so, how important is it? So we had, at this time, of all the images that we received, so we received 19 different MRIs of our 34 patients. And we looked at the first MRI for all of these families. And we wanted to compare the first MRI to 2 patients of the same age who have had MRIs reported as normal, because we wanted to look at 3 specific things: white matter abnormalities, atrophy or changes in brain matter size, as well as the iron accumulation. So there were some important findings that we found here. So we saw that the white matter abnormalities, and the white matter, as you can see on this MRI, is actually this part here that you can see that's slightly white. We see that it tends to happen in this location, which is called periventricular, and deep white matter as well. And we also found that this tended to happen at a median age of 6. The earliest we found it was at the age of 4. The next thing, when we looked at atrophy, we found that in children, the atrophy seemed to be more in the back, which we knew was the cerebellum, or globally, or in this part, which is called the corpus callosum. And this tended to happen at the age of 7 on average. And we do see atrophy in other children when we look at these things blindly. So we had 2 neuroradiologists look at all the scans. And what the only information we'd given them is the age of the patient. So because as age changes, we know that these findings will change over time. And so we know that for example, an older person at the age of 60, 70, or 80 is going to have atrophy on their MRI, but we don't expect it as much as in a child. So we wanted the radiologist to look at it just based on age. We did, obviously couldn't blind everything because we were getting international MRIs done. So there were some things that, said this might be a patient versus this might be a control, but this was out of our, we couldn't do anything better. We tried our best to blind it as much as possible. And so atrophy was important, as I said, in these places, but it seemed to happen after the white matter changes were seen. And then the iron. So the first message here is that most patients did not have iron sensitive imaging, and all of our patients that were controls did have iron sensitive imaging. So what that means is that there are certain MRI sequences that will show iron more readily than our regular sequences. And at our institute, we do these sequences on everybody. But when we were getting our international scans, most of the scans weren't having iron sensitive images. And if we looked for iron on those scans that didn't have iron sensitive images, not a single patient could you identify iron in. So one key message here is if we're looking for iron, we need to look for it in the right image, otherwise we're going to miss it. And so in those that did have iron accumulation, there were only three patients that had iron accumulation seen. And the median age for this was the age of 8. And we did have one family where we could see iron accumulated over time. So when we did see it, we saw it in this area called the globus pallidus, and we saw it in another area called the substantia nigra, which is similar to our other NBIAs. But as I said before, it wasn't prominent, but it could have been missed because it wasn't being done on the right scans. So white matter findings are first, followed by atrophy, followed by iron accumulation, if we can see it. So the other part of this is we were looking at different gene mutations. So FA2H is, a medium-sized, it's a protein that's in this place called the endoplasmic reticulum in the cell. And it's made up of 372 different amino acids that then code for the protein FA2H. So this is an image taken from a website that shows that it goes in and out. There's a segment that's outside, you can see in my arrow, that's called cytochrome B5. There's a segment that goes in and out, and that's another transmembrane protein. But I'm going to show you more details because we have as to why this is important. So let me take you a step back. So in myelin, which is the coating of our nerves, we know that there's a lot of protein and we know that there's a lot of fat. And we know that this FA2H is involved in the fat or the lipids. And we are trying to understand better as to where there is a change going on. We don't have this answer clearly yet. So we know FA2H is involved in this pathway that's going from a fatty acid to this fatty acid that's being hydroxylated, meaning that there's this group called OH that is being added by FA2H somewhere along this pathway. And I say somewhere along this pathway is because we actually don't know exactly where it's being added. And once it's added, then these special types of fats can be, these sugar/fat molecules can be then put together and put into the myelin, which is the layers that are, and help it stabilize and allow for proper conduction to happen in the nerves. We also know that there's certain regions in this gene that are quite the same, no matter what you're looking at. If you're looking at a yeast versus a monkey versus a human. So this is interesting that this gene was actually found in a yeast model way before it was found in humans, and then it has been found in the mouse model. And so I'm going to go back into the mice studies in a bit, but just giving you on that background, what we did was we looked at all the gene mutations and where they may sit. So this is the gene FA2H if we laid it out flat in a line, but we know that is not what's happening in the actual cells. We know that it's convoluted and goes in different ways, and there's certain areas that are probably more important in this gene and in this protein. So we look to see what are the ones in this row that are here closer, these are the ones that we found in our study. And this is all the ones that are bolded above are the ones that were found in previous studies. So we've got all the different mutations here. And so I'm collaborating with Dr. Messias, who is an expert in structural biology in Munich, Germany. And she has kindly agreed to move forward with understanding FA2H further and knowing where the mutations are happening and then the functional consequences of these mutations. So she has built a structural model, which there was a crystal structure done in 2015. And she's looked at this model and she's created her own model of how FA2H would look like in the cell. So this is looking at this place where it sits in the endoplasmic reticulum. And this is on the outside and on the inside. And this part is that first part I told you, which is an active area called the cytochrome B5. And then there's another area where it's just called the hydroxylase region, which is located in a different region here. So again, just in a different way of looking at it, this is one active site, and this is the other active site, the cytochrome B5 and the hydroxylase. So right now, we've just put all the mutations found in our study, and we put it into to see all along the gene, where could it sit when it's actually sitting in its actual conformation. So when we put all the mutations that had a spelling error, so the protein's produced but it's not active, found that the majority of them were sitting in one of the two active sites. And as you can see, these active sites are quite close together. And this is another part of the puzzle nobody has solved yet, is what are they doing? How are they talking to each other? How are they important to each other? So taking these apart, this was the cytochrome B5 region, just showing you a close-up of where these mutations are sitting. And then showing hydroxylase region and showing where the mutations are sitting. And this is the active site again. This is very preliminary work right now. This is, all the mutations are going to be put in it. And then after that, we're going to be looking for funding to try to understand the consequences to the protein itself. There are also some mutations that actually make such a spelling error that they actually don't produce a protein at all. So they stop production altogether. And these were found in both the cytochrome B5 as well as in the hydroxylase domain. And then there were some where the spelling error just caused it to be a frame shift, meaning it kind of moved the whole code over. And so this also causes the protein not to be produced. And this was found in one patient in the cytochrome B5 domain. And same with deletions. So deletions can also cause a problem where the protein is non-functional, meaning there's a chunk that's missing. And these deletions were found in the cytochrome B5 domain or in the hydroxylase domain. So this is what has been done to date, and I want to go into what our next steps are in FAHN research. And this is going to be taking many different directions, but in parallel, so that hopefully we can all collaborate and work together and not wait for one thing to be done before the next step is done. So these are the things that are missing. We still don't know where or what the clear function of FA2H is. We have some ideas, and I'm going to show you some of the ideas. We don't know whether the type or location of the gene mutation matters, like what is actually happening when these mutations are sitting in these different areas. Please excuse the last one, sorry. We don't have an easy biomarker to follow. So we don't have a blood sample or something that we can just mark and follow to see where things are going. We need to bring the mice back into the lab. We don't know how this FA2H interacts with other proteins. Maybe there's some other proteins that can help FA2H work better, and we need to understand this better. And we don't know what the role of iron is. And right now we do still think that it's more of a consequence rather than an actual issue, primary issue. So one project that has been funded and is going to be finally run, there has been a lot of issues getting our control samples more than our patient samples due to COVID, because we've needed to have normal controls to do the study, and all of our ORs were shut down during COVID, and we had to, so they're opened up again, and we're just waiting for one more, which hopefully will happen in the next couple of weeks, and then we can run this study in the summer. So basically what's being done in this study is we're looking at a very complicated picture here of all the different lipids and how they interact with each other. And so I'm working with a colleague, Dr. Stephanie Bennett, who has a platform to measure different lipids through her mass spectroscopy platform. And she can specifically look at FA2H and its consequences of all the lipids around it. So what's being done is she's looking at both skin samples and blood samples and comparing one patient to two controls, and we're going to be that are age matched, because these things could change developmentally, to see if we can find upregulation or downregulation of other areas that maybe serve as a marker for either progression or for disease onset or anything of that sort. Because it would be nice for us to have a simpler way to actually identify different steps that would be very objective. In terms of the mouse, so I'm going to give you a little bit of background on this. So FA2H is highly expressed in the human brain, and this has been seen through mouse models. So this is FA2H, and this is a control of a different kind of a muscle enzyme. And so in the brain, and also in some other regions, FA2H seems to be expressed. And the original mouse, which was a knockout model of a certain type of protein in this pathway, mice were abnormal when this OH group wasn't present, because this then specialized sugar/fat molecule couldn't be incorporated into the myelin. And myelin is kind of layered. It's layered into different sheets, and these sheets have to be quite compact. And so it doesn't seem to be, from the mouse studies that I'm going to show, that it's important at birth, because myelin seems to be present, but what seems to needed is that the myelin has to be stacked appropriately and compacted appropriately in order for things to be developmentally normal. So FA2H, looking at a mouse, seems to increase with time, not at birth, but within the first 20 days, which is equivalent to about the toddler age, if I'm correct, in a mouse. So there have been a couple of mouse models looking at what happens when you just remove FA2H completely from all the cells. What was found was that there were problems in both the central nervous system and the peripheral nervous system, so that the nerves going from the spinal cord to the muscle, and there wasn't any hydroxylation happening, meaning this OH group wasn't being put into into the myelin. And the mouse model that we're going to be pursuing further is this mouse model. And what was interesting about this study is that there were two mice models. There was one where FA2H was taken out of every single cell, and there was another where FA2H was taken out of the cells that produce myelin. And so what was found was that when it was taken out of every single cell, there were problems with movement. There were problems with gait. There were problems with ataxia, things like this that you could see in the mouse. But when you took it out just in the oligodendrocytes, so these are the cells that produce myelin, there were still problems in learning. And there were not problems in learning. They were only problems in cerebellar function, which is telling us that there's something else FA2H is doing outside of the myelin producing cells that is involved in learning and memory. So there's more to FA2H than we think here, in terms of it not just being involved in myelin.
Sunita, I'm sorry, we're starting to run out of time for
Okay, Dr. Remy. Okay, I'm done. I apologize.
sorry.
I'm the last slides. Yeah, I totally apologize. Okay, so Dr. Rahim and his group are actually starting a collaboration with us in terms of development of gene therapy for FAHN. And gene therapy, obviously, as we all know, is exciting hope, where it can be defined as the delivery of nucleic acids to the body for therapeutic benefit, to hopefully deliver a full FA2H gene to compensate for the defective version. And then a viral vector is trying to be produced right now that would be safe to incorporate the FA2H gene into cells. And then this needs to be tested in the mouse model that I said before. So I'm just going to take you through. You're going to hear all about this study from Dr. Lowe now. We are doing a surveillance because we want to find as many FAHN patients as possible across Canada. And we want to continue looking at our patients in the future prospectively. And last but not least, we are going to be looking into caregivers of patients with FAHN and NBIAs as well. So I wanted to end with a video, but I'm going to show the video in the panel, because it's a special video to me, but I will show it to you in the next hour. So I want to just thank everybody, and I want to let Dr. Lowe get his talk going. Thank you very much.
Good morning, ladies and gentlemen. It's my pleasure to give you a talk about our research work. The title of my topic is Patient-Specific Induced Pluripotent Stem Cells as a model for studying fatty acid hydroxylase-associated neurodegeneration. In this talking, I will first give you a general information about FAHN, especially the difference between patients and the mouse model. and also describe the strategy for building IPS model and isogenic lines. Then go through with the generation of FAHN specific IPS models and isogenic lines. At last, discuss using IPS models to study mechanism of FAHN. As you know, FAHN is an autosomal recessive disease caused by mutation of FA2H gene. This disease is associated with nucleotrophy, SPG35, NBIA. The neural degeneration is brain iron accumulation. The clinical characteristics are complicated depending on the different type of mutations. In general, the age of onset in most patients is less than 5 years old. And they have corticospinal tract problems, for example, spasticity, and a mixed movement disorder like ataxia dystonia. And some patients also have eye problem. Late in the disease course, the patients can have intellectual impairment and seizures. The MRI findings are useful for diagnosis. They contain, for example, white matter change, hyperintensity of globus pallidus, pontocerebellar atrophy, thin corpus callosum, and iron accumulation. FAHN is a subtype of NBIA. Some FA2H mutation, for example, these two types of mutation, can cause abnormal iron metabolism, resulting in iron accumulation in the brain, especially in globus pallidus and the substantia nigra. The reason of the iron accumulation in the brain is still not so clear. Some data suggest that the change of intracellular ceramide pore composition, for example, hydroxy fatty acid containing ceramide and non-containing ceramide, the ratio between them is one reason. And also impaired palmitoylation of transferrin receptor is another one. Still, iron accumulation is a cause of neurodegeneration or a consequence of neurodegeneration is still not so clear. Animal disease model is a powerful method to study human disease. Until now, 2 FA2H mutation mouse model has been generated. The first one is null mouse model, in which FA2H is deleted in all cells. And another one, conditional mouse model, in which FA2H is deleted only in oligodendrocytes. Compared to mouse models, the CNS phenotypes between them are similar. For example, demyelination, Purkinje cell loss, cerebellar dysfunction can be found in both animal models. However, learning memory defeats happens only in null mouse model, but not in conditional mouse model, suggests that oligodendrocytes are not responsible for behavior defects. Although mouse models show manifestation of patients, but there are some difference between them. For example, the age of onset, as I mentioned before, usually is less than 5 years. However, demyelination happens at aging mouse at 12 months of age. Furthermore, At early stage in the mouse model, no obvious defect in neurons of oligodendrocytes and astrocytes were found. Therefore, it is necessary to build iPS-based FAHN disease for the study of the mechanism of FAHN. Because until now, the pathophysiology of central nervous system seen as in FAHN is still not so clear. Therefore, patient-specific iPS are excellent alternative to study precise function of FA2H and the mechanism of FAHN. Thank our patients and feminists for the support. We have already got 2 different types of mutations. One is here, another one is here. This fibroblast, we will use them to generate iPS. But keep in mind, there is still a limitation of this patient-specific iPS approach. The main problem is lacking isogenic control because of difference of the genetic background between the patient and the healthy control. And this difference between them will affect the accuracy of the study. Therefore, it is also necessary to build FAHN isogenic iPS lines. We establish isogenic lines from healthy donors using CRISPR-Cas9 technology to create the non-FAHN disease mutation, for example, like this one. Because this type of mutation is associated with nuclear atrophy, SPG35, and NBIA. Therefore, combine both patient-specific disease and isogenic iPS will help us understand the mechanism of FAHN. This figure shows iPS-based disease modeling. The fibroblasts from patients are cultured by in vitro system, and then they will be reprogrammed by transduction with transcription factors and drive them develop into patient-specific iPS. After characterized of this iPS, then they will be differentiated into disease-affected cell model. In our case, like neurons and oligodendrocytes. As a disease modeling, then the pathophysiological difference between patient and the control cells will be detected. In this case, then we can get ideas about the function of mutant gene and the mechanism of the disease. Also, this iPS system can be used as a drug screening by new drug development for the treatment, and also can be used in regenerative medicine. This panel shows you the generation process of iPS. But still, I should mention here that iPS generation is a routine work in our lab. We have already got the FAHN iPS cloning, but by some reason I cannot show you the pictures. Instead, I show you the Niemann-Pick type C1 iPS pictures published from our literature. So go back to the generation process of iPS. The human fibroblasts are cultivated by in vitro system. This is the cells. And then reprogram with transduction by SOX2, OCT4, and so on, and drive them to develop into IPS. And then they form IPS colonies, just like this. After characterization of this IPS, for example, pluripotency, descent, karyotype, mutation type, and the differentiation potentials, then they will be drive to develop into disease-affected cell types, for example, like here. This figure shows you the assessment of pluripotency in IPS. Immunostaining is performed to identify whether IPS colonies express pluripotent markers. This figure shows exactly that the IPS colonies can express LALOG, OCD4, and SSEA4 pluripotent markers. And also, the descent, karyotype, and mutation in IPS should be also proved, as I mentioned before. The descent of IPS is proved to identify whether genetic fingerprint of IPS is the same to the fibroblasts to exclude contamination with surround cells. And also, the karyotype of IPS should be proved to exclude chromosome aberrations occurring during reprogramming. And the mutation type of IPS, of course, should be also identified by sequence to ensure that conservation of this mutation is same to the fibroblast. And also, the differentiation potential in IPS should be also proved. After differentiation, immunostaining is performed to identify whether IPS can differentiate into distinct type of cells. In these figures, the IPS can exactly show that they express nesting, the marker of ectoderm, and also muscle actin for the mesoderm, and alpha fetoprotein for endoderm. The last part of our research is using IPS model to study mechanism of FAHN. After IPS differentiate into desired disease cell types, the difference between the patient's and the control IPS derived cells will be checked in order to find out the precise function of FA2H and the molecular pathophysiology of FAHN. In our case, we drive this FAHN IPS into differentiate into neurons, oligodendrocytes, and astrocytes. Then in neurons, we will compare the difference between the FAHN patient's neurons and the control neurons. And we focus in neuronal proliferation, migration, and differentiation, and also neuronal structure, for example, the axons and synapse, and also some structure. Also the functional property and the synaptic transmission. In this case, when we compare the difference between patient's neurons and control neurons, then we can get ideas about the problem of this disease and get exactly the mechanism of this disease. Of course, same for the oligodendrocytes. We will focus on the difference between oligodendrocytes proliferation, differentiation, and myelin formation. Because in FAHN, the myelin formation problem or demyelination is a bigger problem in both patients and animal models. Also, for the astrocyte, we will focus on the morphology and the activity of these cells and try to find out the difference between the patient's astrocyte and healthy control astrocyte. In this case, get ideas about the disease problem. Because the FA2H is involved in the synthesis of 2-hydroxy fatty acid synthesis. In this case, the membrane lipid metabolism and the lipid buffers in different type of cells, for example, neurons, should be also investigated and try to find out the difference between them and get ideas about the mechanism of the disease. We hope these findings of this experiment will help us understand the disease, the precise function of FA2H, and the molecular pathophysiology of FAHN. Unfortunately, we still haven't started this part, although we have already got the cloning. Because of COVID-19 pandemic in last year, and there is some delay of our project. And I'm sorry about that. We hope that we can report the findings of this data in detail next year. At last, I would like to thank our patients and families for the support and the donation of the cells. And also thanks my colleague, Dr. Andrea Hillman, Dr. Maurice Frisch, and our PhD student, Fatima Effendig. And also thank our cooperation partner, Dr. Venkateswaran from University of Ottawa. And also thanks NBIA Disorder Association for the financial support. And also thanks for your attention.